is made of nucleotides, the building blocks of genetic information. Each nucleotide consists of 3 parts: a phosphate group, a 5-carbon sugar called deoxyribose, and a nitrogenous base. The sugar and phosphate form the backbone of the DNA. Nitrogenous bases are divided into 2 groups, purines and pyrimidines. Purines, which include A, adenine, and G, guanine, have a double ring structure, making them larger. Pyrimidines, including C, cytosine, and T, thymine, have a single ring structure, making them smaller. In DNA, a purine always pairs with a pyrimidine to maintain the uniform width of the double helix. Adenine pairs with thymine, and cytosine pairs with guanine.
DNA's double helix is stabilized by hydrogen bonds between complementary base pairs. Adenine forms 2 hydrogen bonds with thymine, while cytosine forms 3 hydrogen bonds with guanine. These hydrogen bonds allow DNA to unzip during replication and transcription. The 2 strands of DNA are complementary, meaning that one strand's sequence determines the sequence of another.
Prokaryotic DNA exists as a single cellular chromosome found in the nucleoid region. Because prokaryotic cells lack a nucleus, their DNA is not enclosed in a membrane. To fit inside the small cell, prokaryotic DNA is supercoiled. Enzymes twist the circular chromosome into tight loops, making it more compact.
Eukaryotic DNA is much larger than prokaryotic DNA and must be tightly packed to fit inside the nucleus. First, DNA wraps around the histone proteins, forming nucleosomes, which look like beads on a string. These nucleosomes coil into chromatin fibers, which further fold into thick, compact structures. During cell division, chromatin condenses even more to form distinct chromosomes.
Mutations occur when there are changes in the DNA sequence. These can be caused by errors during DNA replication, exposure to UV radiation, harmful chemicals, or even viruses. While some mutations have no effect, others can alter proteins, leading to changes in an organism's traits.
Point mutations occur when a single nucleotide is changed. The effects depend on how the mutation alters the genetic code. Some point mutations cause no harm, while others can change the structure of a protein, and some can code for a stop codon. Silent mutations is a type of point mutation where a single nucleotide is changed, but the amino acid sequence remains the same, due to the redundancy of the genetic code. This means the mutation has no effect on the final protein.
A frame shift mutation occurs when a nucleotide is added or deleted from the sequence, shifting the reading frame of the genetic code. This alters every codon that follows, often resulting in a completely non-functional protein. For example, deleting a single nucleotide in a gene that codes for a protein can drastically change the amino acid sequence.
Transposable elements, known as jumping genes, are DNA sequences that can move from one location in the genome to another. Some transpones cut and paste themselves into new locations, while others copy themselves before inserting elsewhere. These movements can cause mutations by disrupting genes or changing gene regulation.
Chromosomal mutations involve large changes in chromosome structure. These mutations can include deletions, where part of a chromosome is lost, duplications, where extra copies of a gene are made, inversions, where a section of a chromosome is flipped, and translocations, where segments of different chromosomes are swapped.
Conjunction is a process where bacteria transfer genetic material directly to each other through a pilus, a tube-like structure. One bacterium with a special plasmid, called the F plasmid, extends a pilus to another bacterium and transfers genetic information. Transduction occurs when a bacteriophage, a virus that infects bacteria, accidentally transfers bacteria DNA from one cell to another. During infection, the virus sometimes mistakenly packages bacteria DNA instead of viral DNA. When the virus infects a new bacterium, it injects the previous bacterium's DNA, potentially giving the new cell new genetic traits. Transformation happens when bacteria take free DNA from their surroundings. This can occur when a bacterial cell dies and releases its genetic material into the environment. Nearby bacteria can absorb the DNA and integrate it into their genome.